Hello, my name is Thomas Han, and I'm CEO and co-founder of PEROXITECH. We are a biotech startup out of the University of Pennsylvania. Today, I'm going to spend a few minutes to present our lead program for the treatment of COVID-induced acute lung injury. As we know, acute lung injury, or ALI, is one of the primary causes of death in COVID patients. This is occurring due to the uncontrolled immune response and resulting damage caused to the lungs. Unfortunately, the prognosis for COVID-ALI is poor. Latest data show the mortality for patients with COVID-ALI to be approximately 60 to 80%. There are no approved therapeutic treatments available. Based on our clinical study results, our lead candidate is projected to improve mortality outcomes, reduce severity, and decrease ventilated days, leading to shortened hospital stays. Our lead compound, PIP2, is a 9-amino acid peptide derived from human lung surfactant. It will be dosed IV for short-term acute therapy in the ICU setting. In the following slides, I'm going to review the breakthrough scientific discovery made by our co-founder and scientific lead, Dr. Aaron Fisher. So what's going on with COVID and ALI? We now know that for about 20% of COVID patients, their immune response becomes uncontrolled and leads to catastrophic lung damage. The primary pathway for COVID-ALI is the overproduction of reactive oxygen species, or ROS. In healthy patients, ROS is an important component of our immune response. However, when patients are infected with COVID, production of ROS becomes uncontrolled and leads to a cytokine storm. Overproduction of ROS causes inflammation in the alveoli capillary barrier, leading to lung edema and rapid decrease in blood gas exchange. The end result is respiratory failure. Prior to Dr. Fisher's pioneering research, the primary mechanism of action in the lung was thought to be driven by RAC1 and cytoplasmic factors, leading to ROS activation. Dr. Fisher discovered an earlier pathway for ROS activation via a new protein, PRDX6. PRDX6 is one of the earliest pathways identified for RAC1 activation. We then discovered a novel drug that inhibited PRDX6. Our lead compound, PIP2, is a 9-amino acid peptide that is based on human lung surfactant. Substance studies showed PIP2 binds to PRDX6 and disrupts the ROS production pathway and can potentially protect lungs from damage. Funded by NIH grants, we completed several studies for ALI using PIP2. This was a 5-arm, 24-hour ALI study using lipopolysaccharide, or LPS, to induce ALI. LPS is also accepted by the FDA in human trials to model lung injury as well. The control arm was healthy normal mice with no LPS, placebo, or treatment arm. For the rest of the arms, LPS was dosed at the start of the study. Arm 2 received placebo. Arm 3, 4, and 5 all received PIP2. The difference was the timing of the PIP2 dose. In Arm 3, PIP2 was dosed at the start of the study. This was done to model prophylactic treatment. In Arm 4 and 5, PIP2 was dosed 12 hours or 16 hours post-LPS dose. This delayed treatment arm represented the typical clinical setting where diagnosis occurs following ALI symptoms. On slide 9, the results of the study were better than we had expected. The first bar in white is the control arm. This is normal, healthy lungs. The next bar, in solid black, shows the LPS arm without any treatment, and as expected, there is significant lung damage, including ALI. The next bar with horizontal stripes shows PIP2 dosed prophylactically. As you can see, there's almost no lung damage. It is comparable to normal lungs in the white bar. In this scenario, PIP2 prevented the onset of acute lung injury when dosed prophylactically. The next two arms are the 12 hours and 16 hours delayed treatment arms. In both arms, PIP2 demonstrated rescue of damaged lungs back to near normal function. These results clearly show the validity of the new PRDX6 drug target and PIP2's ability to protect and rescue lungs from ALI. In this 5-day ALI mortality study, the dose of LPS was increased 3 times versus the previous 24-hour ALI study. The LPS was instilled intratracheally in both placebo and treatment arm at the start of the study. Then 12 hours later, the placebo, or PIP2, was respectively administered via IV. Subsequent dose of placebo or PIP2 was repeated every 24 hours for 5 days. Then all mice were sacrificed on day 6. Slide 12 shows the dramatic results of the study. 71% of PIP2 treated mice survived versus none in the placebo arm. In our research, we have not seen any other treatment demonstrate these levels of mortality improvement in ALI. Our most recent study is a 5-day sepsis-induced ALI mortality study. Due to the systemic nature of sepsis, this is considered one of the most challenging ALI studies. In this study, we added a high-dose arm of PIP2. We wanted to see if there was a dose response and to see if a higher dose would result in any significant adverse events. In total, there were 3 arms, 1 placebo and 2 treatment arms. The low-dose PIP was identical to the previous ALI mortality study. The high dose is 10 times higher at 20 micrograms per gram. Since this is a sepsis study, the LPS is administered intraperitoneally at the start of the study. Then placebo or active was dosed 12 hours after the LPS. Additional doses of placebo or active were given at 24-hour intervals for 5 days. Then any surviving mice were sacrificed on day 6. Slide 15 shows the groundbreaking results. 70% of the high dose and 38% of the low dose PIP2 treatment arm survived, versus none in the placebo arm. Also, there were no observable adverse events in any of the treatment arms, including the high dose arm. Sepsis-induced ALI would represent a worst-case scenario in COVID-infected patients. With these type of efficacies, PIP2 could significantly reduce mortality in COVID patients. In conclusion, with acute lung injury driving the majority of COVID mortality, our lead candidate has demonstrated compelling results. In the 24-hour ALI study, PIP2 prevented lung damage when dosed prophylactically. Then when dose is delayed up to 16 hours, it was able to rescue damaged lungs to near normal function. We have also demonstrated superior mortality study in 2 separate outcome studies. In both cases, 70% of PIP2 arms survived versus none in the placebo arm. Based on these results, we project PIP2 to provide several clinical benefits for COVID. Importantly, improved mortality outcomes in COVID ALI, decreased need for ventilation, and reduced duration of ventilated days, leading to shortened hospital stays. With these kinds of results, PIP2 would be a game changer in our battle against COVID. PEROXITECH is in our Series A funding window, and we look forward to continuing our conversation in greater detail. Thank you.